Well, it's so nice to meet you, Dr. Matheny. Do you want to tell us about your work?
Yeah, it's been many years in the development, and I am by training a cardiac surgeon. Practiced in Indianapolis for 8 years and then moved to Atlanta. During my time in Indianapolis, I did some research at Purdue University, and that is where they came up somewhat accidentally with the extracellular matrix um, having an influence on regeneration. And the extracellular matrix is everything outside the cell. So you're made of two things: you're made of cells and you're made of extracellular matrix. And for the longest time, every biologist has emphasized the cell and the complexity and the importance of the cell, which is true. Um, but not a lot has been given to the attention of the extracellular matrix. When I was in medical school, it was called the ground substance, and we thought, oh, it contains a few factors and it contains collagen, and basically it just supports the all-important cell. And now, over the last several decades, we've learned that the extracellular matrix is as complex as the cell is, and the two are in this, as Mina Bissell described in a very important 1982 paper. The two are in a state of dynamic reciprocity, and that is a continuous back and forth informational exchange between the outside of the cell and the microenvironment across the cell membrane to activate either conduction pathways that transit to the nucleus or the actin-myosin cytoskeleton, which transits information to the nucleus that convert those signals, either chemical or physical, into an epigenetic alteration to express the genes that are necessary for proper phenotypic expression of the cell in that location. So all that is to be said is the extracellular matrix tells the cell what to do. And like a computer, you need a hardware and you need software. So the information is passed mechanically: stress, strain, compression, flow disturbances from the outside of the cell to the cell, so it can then make the proteins that are necessary for that environment. An example of that is what we call Wolff's Law, where the tuberculations in your long bones actually line up against the forces of gravity. And if you don't have gravity, those tuberculations will actually start to disappear or dissociate. So the forces actually dictate what the structures become. And we call this form follows function. Doesn't expand the genetic code, but within the constraints of the genetic code, the organism is adaptable to some extent. So the corollary to Wolff's law is Davis's law in the soft tissue, where if you have stress and strain and pull, the cells will recognize that because of their attachments to the extracellular matrix. That information gets transmitted in a lock and key fashion that's very specific for the proteins to the nucleus, where epigenetically the genes are manifest to be transcribed to deal with that microenvironment. And it's not that sounds complex, but if you have a dumbbell and it weighs 50 pounds and you keep lifting it, your muscle's going to get bigger, okay? Because the forces are required and your body adapts within some constraints. This is not a genetic change, it's not a mutation, which, just as an editorial off to the side, being a clinician, it's easy for academics and some biologists who look at genetic change to infer that that is a Darwinian expression of the mutations to change. In real life medicine, we call that disease. Okay. You have genetic disease. You change one amino acid and you get problems with cystic fibrosis, you get thalassemias, you get different types of a myriad, hundreds, thousands of different genetic diseases when you have mutations. It's a problem. It's not a benefit. So, all of that was very interesting when academicians present that, because I'm going, all we see is disease. When you have a genetic mutation, the organism gets weaker, not stronger. Um, It doesn't diversify. But going back to the science of the extracellular matrix, this was used basically accidentally to begin with, and then purposefully to provide a microenvironment for the cells to function in, and we can make products out of that. So some extracellular matrices, or ECMs, are quite proliferative. Some cause angiogenesis or new blood supply. Some are what we call immunomodulatory. That is, that they actually change the default mechanism of scarring to one of regeneration. Some animals already do this: planaria, starfish, axolotls, zebrafish. You can give them an infarct. You can do different, cut off their arms even, and they'll grow them back. And that's because the matrix actually directs that cell differentiation after the cells dedifferentiate. So, but humans and mammals, we don't heal that way. We heal by scarring. And our immune system is inflammatory. So you have to locally alter that default change and response of the immune system to one that's non-inflammatory. And then those genes are capable of expressing the proper tissue at that location. So we take the matrix, which there's different varieties and kinds, but the one that we use the most is called SIS, or small intestine submucosa. And the small intestine submucosa is very proliferative. You actually replace the lining of your intestine about every 7 to 10 days. So new cells have to replace that. So the basement membrane and the submucosa are very proliferative in their signaling for the cells to divide and grow. It's also immunomodulatory in that it keeps inflammation from occurring very much in the manner that the placenta does for the mother and the baby. They do not have the same genetic code. So the baby would reject mother if the placenta wasn't there. As it is, the children usually wait till they're teenagers to reject mother now. So that's there's no help with that. But that type of immunomodulatory capability is with the small intestine as well. So we can make that into different products that go into the body that provide the proliferation, the angiogenesis, and the immunomodulation so that those tissues get regenerated rather than scar tissue formation. So in medicine, in surgery especially, in cardiovascular medicine, everything that we use right now is decades old technology, Dacron grafts, PTFE or Gore-Tex, which in my opinion should never be used in the body. It should only be used as raincoats. So we want to get away from the synthetics that have inherent problems, inherent scar formation, and never turn into the tissue. This is of great benefit to the patient, but it's particularly valuable to the pediatric population because none of those synthetics will grow with the babies. Mm-hmm. So just I was telling Sal before we left the meeting, we had a case coming up the day before yesterday at Cincinnati Children's. We replaced a heart valve in a 4-year-old. Well, that baby had had 4 previous operations for synthetic valves that either wore out or clotted off and didn't have any capability of growth for that child. So we put in a heart valve made out of the matrix, and this may be the last valve that child needs because it will turn into their tissue when over the next several months, the implant actually goes away and you're left with their tissue that functions as normal tissue, a normal design of the valve, and is capable of growth with the children. So this is really where we're going, is taking some of these new discoveries in the extracellular matrix that are capable of regenerating tissue and making different products out of that, so we can really usher in a new era of medical devices that aren't just engineered from synthetic constructs that all have their shortcomings to something that is much better and is of the patient's own tissue, not a synthetic. So that's
That's
kind of
amazing.
that's kind of the whole story in a summary. Uh, there are different companies around the country, around the world that are using the matrices, starting to augment and change the matrices so that when we find those factors that are providing those benefits, we can actually increase them within the matrix and augment it so we get an even better effects. So that's the direction that the technology is taking.
The video that you sent me, is it for public release? Was that you operating on the little girl?
No, I was instructing on how to put the valve in, and the surgeons there pretty much know how to do that. It's not by any means outside of their current skill set, but I like to be there in case there's any questions. So I'm there for every operation till these guys have done a half a dozen of them and trained to put it in just in case any nuances. You know,
So,
you as long as there's no identifying factors of the institution or at least till I get clearance or the patient, you can show this. Really, to the untrained eye, all this shows is what heart surgery in a baby looks like. You can see how complex it is. Um, you know, this is what has to be done. So you really don't want a child to have to go through this more than once. And the fact that as they grow, they have to go through this 2, 3, 4, 5, 6 times is a tragedy. So we would like to get it to the point where one and done. That's what I like to say.
so you were able to reprogram. So maybe we could you could describe a little bit more, and our audience is usually very sophisticated. Um, my understanding then is you're helping to you figure out a way to reprogram the cell so that it will grow. What was the specific thing that they were replacing here?
A tricuspid heart valve. You
A
have
heart valve.
4 valves in your heart. Mitral and tricuspid are what we call AV valves, atrioventricular, and then you have your aortic and pulmonary valves, which are semilunar cusp type valves. So this was the tricuspid valve that we were replacing here. And after we do the tricuspid and get that on the market, then we're already starting to study the mitral valve and the other 2 valves as well.
So do you actually grow the valve like in a outside and then implant it? You take the patient's cell. Is that how it's?
No. The traditional tissue engineering approach, which is now 40 years old, you would go and find your cells, and then you would get your scaffold, and you would create the scaffold in the 3D architecture that you wanted it to be, such as a valve, and then you would put the 2 together in a bioreactor, and the cells would adhere to the scaffold, and that would make the valve, and then you would take it out of the bioreactor and go put it in the patient. All of that is very interesting science, and there are thousands of articles on which type of cells to use. There are thousands of articles of which scaffold to use. There are thousands of articles of bioreactors and how you make that. The problem is, after 40 years, we never use any of that stuff because it doesn't really work. Okay. So there is nothing in cardiovascular surgery that is created that way that's approved right now for use. And part of the problem is the first thing of those 3 things I mentioned, that you have to have angiogenesis, proliferation, and immunomodulation. There's no blood supply. Nothing in the body works without a blood supply. So you take something that's tissue engineered out of the bioreactor, it's lost all of its nutrient. And there is no blood supply. So I remember I used to be excited 10 years ago when people would show me heart cells in a petri dish that were beating synchronously. And they would say, "This is great. We're going to make a heart patch." And I said, "No, you're not, because 6 minutes after you take that out of the bioreactor, it's going to be dead because it has no blood supply." And that's the way we normally grow, is you get the blood supply and then the tissues develop. And that overarching theme is kind of inherent to our devices, is let's let the body do what it has already done before. We have all made our valves, we've all made our spinal cords, we've all made our bones, and now they need to be replaced. So the information is there. It just needs to be opened up, because a lot of your nascent genes have been basically silenced, because you go from growth and proliferation as a child to an adult, when then the information becomes static and maintenance in nature. You are a liver. You're as big as I want you to be. So I just want you to sit here and do what livers do. I don't want you to grow anymore or proliferate or wig off in some strange direction. So actually, the extracellular matrix to an adult organ changes to contain that proliferation from happening and to limit it. It's very much that way in the heart. So when people were injecting stem cells, I was saying, this is not going to work, because you're hoping that stem cell will turn into a cardiomyocyte and start functioning and proliferating, but that's not the way the heart matrix work. It's very non-proliferative, and every signal that that stem cell is going to get is going to say, don't grow, don't proliferate, you're not welcome here. So that whole area got billions of dollars of investment, but hasn't worked. It did provide a lot of interesting information, though, because what we learned with the use of the stem cells was a whole new area of exosome production. And that is a whole new level of communication within the body and the local area that's going on that we didn't really know was there. So exosomes are these tiny nanoparticles that are contained in lipid membranes that the cells spit out. So any signal of efficacy that you get from stem cells isn't from the cell becoming that kind of tissue. It's from the cell basically acting like a pharmacy and pumping out these little particles that cause proliferation and angiogenesis and immunomodulation. So those same particles we have found, largely to the work of Steve Badlack at McGowan Institute in Pittsburgh, demonstrated these particles become attached to the ECM and are like time-release capsules. As cells migrate into the matrix, they pick up these particles that then instruct the genes to be angiogenic, proliferative, and immunomodulate. So we need to harken back to the embryologic development and let the body activate those nascent genes that were there and active when we were all developing in utero, and just let them do their thing again. So this is called in vivo tissue engineering, not in vitro tissue engineering, because we're not going to put it in a bioreactor. Well, we do, but the patient's the bioreactor. And we don't have to go get any cells, because you've got 30 trillion of them in your body. So you bring your own cells, you bring your own blood supply. We're just going to construct this and have the instructions to re-energize the same events that were present when you were forming this initially. So does that make sense?
Yes, and this leads. We have one more maybe technical question, then we could get into some other areas. Okay. You had, when we first met, you'd related the story that you were presenting at a conference, and there were a lot of advocates of embryonic stem cells, and they sat you down because you were finding an alternative to embryonic stem cells that actually work better. If you could relate that, this would be awesome.
Yeah, actually that has happened on several occasions, but I was, because of the regenerative nature of what we were doing, we were invited to certain conferences. And I remember telling several of my friends, this is this is not going to be well received. And they said, no, this is great. I mean, you're actually accomplishing what they want to do. They're trying to regenerate heart muscle, and they haven't really been able to do it. And you're regenerating heart muscle that's not only a few cells, but full thickness trabeculated ventricle. So they're going to love it. I said, no, that's not how science works. You know, Thomas Kuhn in his Structure of Scientific Revolution will tell you it's not well received by the ingrained priesthood. So I got up there and said, guess what? You don't need to go harvest stem cells. You just put this stuff in and it does it all on its own. And I was asked to step down from the podium because neither the organizers nor the presenters wanted to hear what I had to say. And of course, in hindsight, all of that had great excitement, and then it went away as the stem cells weren't working, and there were dozens of different types. And the thought was, well, we just don't have the right type of stem cell, or we don't have the right concentration of stem cell, or they're just not lasting long enough to work there. And it was none of those. That's not how they work. So I was asked to step down from several podiums that basically, when people are doing research and they've got millions of dollars of funding and you just told them that their baby's ugly, they don't take kindly to it. So, you know, it doesn't always set well.
Well, so when we met, it was really news to me that the all this embryonic stem cell research for regenerative tissue had fallen out of favor. It's still very important to study stem cells for sure, but not in the context of just inserting them into human beings and creating medical cures. So I saw your the website Core Matrix.
Correct.
And so these products are now being used to heal people. If you have any stories you'd like to relate.
Well, once I saw that we were regenerating myocardium, this is not something that's supposed to happen. The cells you get shortly after birth are basically all that you get, with the heart turning over only a few times during your lifetime. So once you get an injury, ischemia, heart attack, that turns into scar, and that's all you get. So the fact that we were regenerating myocardium, which was blasphemous to begin with, actually pointed me towards the ability to possibly cure heart failure, which is the #1 disease entity in the Western world. And so really excited about making an impact on this, because there is no cure for heart failure. You have very expensive but palliative treatments. You have transplants, you have pacer devices, you have ventricular assist devices, or you can give medications, but none of those fix the underlying problem, which is loss of cardiomyocytes. You've lost a big chunk of your heart, and now it's scar tissue, so it doesn't beat. But now we can start replacing that and bringing that back functionally. So we have used the ECM and been trying it in different applications to restore that. The animal studies have gone well, and then we got our first cleared product by the FDA, which is applying it to the surface of the heart where an infarct has occurred, and we're showing beneficial effects there. So that's enormous. And then that was merely our first product that was a sheet of the extracellular matrix. And so several of my more cavalier surgeon buddies started forming that into heart valves. We had planned to go into heart valves, and I first published on that in '99, but they kind of moved our timetable ahead because they were using this off-label and, so to speak, rolling their own. So with their little origami methods on the back table, we're fashioning heart valves in situations where prosthetic wouldn't work, such as the pediatric population that didn't have one made, even in the smallest sizes, to apply to these patients. So we were kind of pushed into the heart valve business too, but that has gone well. So we're in the middle of a clinical trial right now, our pivotal trial for the tricuspid valve, and hopefully we'll continue both direct surgically and percutaneously on a catheter be able to deliver all the heart valves over the next several years. And these will be valves that will regenerate into normal valves, not just biologics that decay or metal valves that have their own inherent problems. So,
Well, we're running up on the end of our show. I know you're a busy man. We are so grateful for your time. So I wanted to explore some topics. Now, I ran into the idea of embryonic stem cells in school, and I was heartbroken when I had to do a report on various studies for as a class presentation on embryonic stem cells, and these were harvested presumably from aborted fetuses, and that really did break my heart. But also in the process of as I was studying this, there was a topic of immortalized cell lines. It seemed that there had been the capacity in human cells to live forever. If you'd like to comment on that. And
yeah, that's been one of the side features that has been fascinating to us. You had mentioned Yamanaka and his iPS cells, which from his work for which he received the Nobel Prize, basically taking a fully differentiated cell, such as a fibroblast or a granulocyte or other cell, and with four specific transcription factors, able to revert that cell back to an embryonic stem cell. Okay. And what he found was those special four transcription factors did what we had noticed, unlocking those nascent genes that had been stopped once the embryologic development had been complete. So you unlock those and the cells can actually revert back. So the cells are plastic, but in that they can be molded different ways, but they are plastic within the constraints of their genetic code. They can't become something else. So then you can take that cell that has gone back to the embryonic state, you can put in different factors and you can then send it down a different lineage. Like you take a fibroblast, revert it back, and then make it a cardiomyocyte, or make it a neural cell, or make it something else, because the genetic code is within that cell to do those things if you take it back to the starting point. Okay, we started, but what has also been noticed with those cells is they become younger. The telomeres, which is a marker of age, which gradually gets shorter and shorter with each cell division, actually grow back out. Telomerase activity goes up, oxygen radical species go down. So when we started looking at what was happening to the cells around our matrix, we started noticing some of the same things. Some of the same proteins started being expressed, indicating the nascent nature of the genes that were being expressed. If you take an adult fibroblast from the heart that is near its end of life and put it on a young matrix, the telomeres grow back out, telomerase activity goes up, oxygen radical species go down. So it tells you that the age right now is unidirectional with us growing old, but it doesn't have to be. And there are other studies that are going on now in different, where you biologically, you can introduce youthful factors into adults and actually reverse the aging process. So this all sounds very science fiction, but we're just on the cusp of that. But what it told me was living long lives as recorded in Genesis is not impossible. In fact, that can very much happen, because as I learned from Walt Brown, those ages in Genesis that decrease after the flood are in a perfect negative logarithmic curve that shows that a new detrimental effect was introduced into the population till you achieved a new steady state. So very exciting to see the possibilities there. And for those that see the film, this isn't my quote, but I was at a meeting where the presenter said, those individuals who will live to be 150 years old have already been born. And that's the type of thing we can look forward to.
Yes, and I just related to my research, I worked for famous genetic engineer John Sanford,
Mhm.
and it was also his conclusion that people had lived long. That seems very much in the realm of possibility. Uh, I know, for example, uh, we have progeria, which is early aging, and the human being, these individuals live like only 1/5 the normal lifespan, and it is only just a few mutations in nuclear lamin A
Yeah,
that will cause them. So it's to me, it's perfectly feasible that some genetic mutations in the past, uh, if they weren't present, we could be living 5 times longer because it could be just that small of a change of degradation.
yeah.
So what we're doing, I think, in the research you're doing, we're finding ways to work around. We're not re-engineering the genome, but we're finding ways to kind of work around the problems that we've inherited.
Right. If Yamanaka can completely revert a cell back to an embryonic state with 4 transcription factors, it may not be that many that were affected that caused the change. That's correct.
And there's one other aspect that's kind of near and dear to my heart. Um, I actually believe that it's very provident. We have model organisms like sheep, etc. That the reason it's very provident we have creatures like chimpanzees that are so similar to us, we can actually run a lot of medical testing without having to actually run this first on human beings. So, what model organisms have you worked with? And then I'm going to hand it over to Rebecca in their remaining time. Thank you for indulging my questions. I know Rebecca probably has some questions, but if you could talk a little bit about model organisms.
Yeah, we typically use small animals and then large animals. The small animals is just to see if the proof of concept is there. And we always have to justify how many animals we use and if there's anything else in the literature that would help us. So we go through those steps of limiting the number of animals, but we'll do it in small animals, which is either a mouse or a rat. If it proves out, we'll go to the next level, which is partially dictated by the anatomy, the physiology, and the regulatory bodies, i.e., for heart valves, the preferential model is sheep because they tend to calcify everything. So if you can have a valve that remodels and lasts a long time, and we've had some of our animals out to 2.5 years, they will typically calcify any of the current bioprostheses in 4 months, and they'll become hard as a rock. So if you put it in a sheep and it doesn't calcify, then you're on the right track. So that model, the valves are similar, and the model is what is required for regulatory purposes.
Wow. Well, Dr. Matheny, this has been amazing. And I'm really curious about, how this relates to intelligent design. And you kind of mentioned the problematic thing about mutations for evolution, but can you talk a little bit more about what you've learned and as it relates to the theory of evolution or with intelligent design?
Well, one of the places this clearly impacts, and we're the rabbit hole is getting deeper and deeper as far as the amount of information exchange and the nano machines that are at work. The external environment talks to the cell through the membrane receptors that are lock and key, and so you can't have one without the other. And people talk about the chicken and the egg. Is it the chicken or the egg? Well, when we're talking about this much information, it's the chicken or it's the egg. it's it's huge because the amount of amino acid proteins that are involved in doing that with each step is myriad, and they all had to occur together. And with a background of in the universe of entropy, you don't get a little protein that stays around waiting 1 million years for another protein. They all had to occur together. And you don't get the adaptability. You don't get adaptability. You don't get homeostasis unless they all happen together. And we're finding that the information bits that are transferred, not only in the DNA, but now with the ribosomes and the messenger RNA and the exosomes is enormous, and it's fine-tuned and it's directed, and that doesn't occur by accident. The only way we know that happens is mind plus energy equals design. So it had to be designed, and anybody that can't see that is not rejecting that data because of the information and the scientific quality. They're rejecting that because they don't want to believe it. It's like Einstein putting in his fudge factor into his equation, recognizing that the implications of a starting point for the universe were not acceptable. So he rejected that and put that fudge factor in it, not because he knew it needed to be there, because he couldn't bring himself to accept the fact until later that there was a beginning when the data became so overwhelming. So a lot of the people that don't believe in intelligent design are not there because the data doesn't not only suggest it, but proves it, but because they just don't want to believe there was a creator, which is unfortunate, because when you know that creator and you recognize that he has that much power, and yet he died for you and your sins, such power, such love, why would you not want to know that person? It's incredible.
Yes. Well, the answer that I hear a lot is, well, we don't think it was that complicated to begin with. It started out as something simpler. So what's your response to that? Can they avoid the chicken and the egg problem with just saying it was simpler?
Well, if they gave me a plausible mechanism, I might believe that. The problem with that is actually one I asked my high school science teacher, and I said, we're discussing entropy, where things fall apart in the universe and go from higher order to disorder, and higher energy to lower energy, and energy is lost. In the background of this entire thermodynamics problem, how can you postulate an increasingly complex system by accident? No, any system breaks down. It doesn't get more and more complex. I don't come home and my house is clean or my car runs better. I have to put energy into making those go in that direction. If you can show me where a system within a breakdown system could actually do that, but that's been the problem with Darwinianism all along, and especially origin studies, is they don't have any scientific mechanism. They're not the ones guessing. I mean, we're not the ones guessing. They're guessing and postulating things that really have never held up. Most of the theories that are out there right now, the only thing that they're lacking is the first statement that says once upon a time. It really is.
Yes. Wow. Well, what about, they are say that, well, most mutations may be harmful or neutral, but every once in a while, these mutations come along that just revolutionize the creature. So in your work, have you seen any evidence of this?
No, it's been the opposite, but that's not what Darwin said, slight successive changes. So there's not one that comes along every once in a while that pops up with a myriad of changes. And Gould tried to explain that away with his punctuated equilibrium. We know that it couldn't have happened by slight successive changes because there's too long a life span between one protein and the next in the beneficial cause. So he said punctuated equilibrium. So by some mechanism that has never been explained, we had 100 changes, and then a while later we had 100 changes, and a while later we had 100 changes. But everything that we see is a weakening of the organism with genetic change, not a strengthening.
Well, we have about 3 minutes left, and we want to honor your time. If you had any, Rebecca, if you had any questions, I'm kind of all talked out. And
well, I'd just like to know, is there anything, Dr. Matheny, that you think is important that we didn't hit on?
Well, I gave you an awful lot in just a few minutes, but I think as we go forward, the science is going to become more and more compelling for the design inference because it's just becoming so complex. It's not this bag of goo that Darwin and his peers thought the cell was. It is so complex, not only now within the cell, but I think the extracellular matrix is an exponent to the complexity of the cell. It doesn't double it, it exponentially increases it because the two had to happen together. And it's thousands of proteins working in stoichiometric perfect harmony that had to be designed.
Well, I didn't have anything else. This was so generous of you. And I hope we meet again.
Yeah, Sal, good to see you again. Enjoyed the weekend. Becca, nice to meet you. And sorry I have to shove off, but some other things to take care of.
Nice to meet you. Thank you so much for your great work.
All
May
right.
God bless you.
God bless you.
Okay.
Take care.
Bye.
Bye.